Welcome to the second part of microbial metabolism. Enzymes may also be classified based on where they act in a cell. Most cellular enzymes are endoenzymes, and they're found to have their action inside the cell. Hence the term endo. And most of these enzymes are metabolic enzymes. So they drive the metabolism of the cell minute to minute, day to day through the life of the cell. And most of the substrates that endoenzymes act on would be described as small substrates. This would include substrates that might be small pieces of food, or like nutrients such as amino acids or simpler sugars, and they can make their way into a cell as smaller substrates as they can fit through the cell wall or cell membranes of bacteria, and then endoenzymes can act on those. However, we know that some substrates, food sources and such, are very large, like complex polysaccharides or very large proteins, and some fats can be very large, and so some enzymes are referred to as exoenzymes because they'll be secreted and they'll act outside the cell on larger substrates. So you can predict that if a substrate or source that's outside the cell is too large to fit through the cell membrane, then an exoenzyme may be involved in then breaking down that substrate extracellularly into simpler molecules that then can be imported into the cell for further digestion. And we say usually exoenzymes act on larger substrates, but there can be other reasons for the production and secretion of exoenzymes. So we'll write down here that exoenzymes are secreted, exo referring to outside the cell, but then another reason for exoenzymes may be that there's something outside the cell that could be dangerous, or the cell may need to deal with before that item would do harm to the cell or be taken in. And so we can talk about neutralization as an event. Some exoenzymes are designed to neutralize a threat to the bacterium in the external environment. And just to use a couple of examples, one from our textbook and one that we learned about in laboratory, the lab one that we learned about was H. pylori. We know this is the causative agent of stomach ulcers. And the way in which stomach ulcers occur is that H. pylori has figured out that it can secrete a urease enzyme, which will neutralize stomach acid because one of the end products of urease is ammonia, and then the dangerous stomach acid will be neutralized. So HCl will refer to the stomach acid. And then another example would be Neisseria gonorrhoeae. The cause of gonorrhea is known to produce an extracellular enzyme known as IgA protease. And IgA protease refers to an enzyme that breaks down an antibody, a defensive protein that is instrumental in trying to defend against Neisseria gonorrhoeae infection. And so what can happen is that the Neisseria gonorrhoeae, this N is not underlining properly, the Neisseria gonorrhoeae organism, this is not spelled properly, here we go, there it is, okay, is not able to usually survive unless IgA immunoglobulin antibody is neutralized. And so the pathogenic strains of Neisseria gonorrhoeae that cause gonorrhea symptoms and sexually transmitted disease are known to secrete this enzyme then that goes into the area that it infects, and then the antibody does not have the proper action because it gets dissolved by this protease enzyme. Types of enzymes may also be classified on when they're produced. One type is constitutive, and this is a technical term that means that an enzyme is found as present on an ongoing basis within a cell. Enzymes that are found as always present in a cell are mainly metabolic type enzymes. Enzymes involved in needs that the cell constantly has, like respiration type events. A second type of enzyme would be an induced or adaptive type of enzyme. This class of enzymes are not continuously needed by the cell, but are only produced when a substrate is present. So the cell has to sense that a new need has occurred in the environment, and then the cell will turn on the genes to make these enzyme products for digestion, and typically these will be produced at a high level but for a short period of time or as needed. And the goal of induced or adaptive enzymes is to save energy, because it may not make sense for the cell to produce these types of enzymes at all times, but to only turn on these enzymes in their production when necessary. One of the most well-known examples in bacteria for such induced or adaptive enzymes would be the lactose operon or lactose digesting enzymes. There's an enzyme called beta-galactosidase that we know can split up the disaccharide sugar lactose. And so beta-galactosidase would only be produced for lactose digestion when lactose is present, which may not be continuously present in a bacterium's environment. So the bacterium has to sense that lactose is there, and then to produce the enzyme beta-galactosidase for lactose digestion when the lactose is present. And then when the lactose is digested or is no longer in the environment, then the genes for beta-galactosidase production will turn off and save energy. Earlier in this lecture, during part 1, we learned that enzymes are primarily made of protein-based structures, the apoenzyme portion of an enzyme. And as a side note, a different class of enzymes has been discovered and described called ribozymes. And ribozymes are different in that ribozymes are described as RNA that can cut and splice other molecules of RNA. And this has to do with something called the spliceosome. In eukaryotic cells, there's a complex known as the spliceosome. And spliceosomes are defined as a collection of small nuclear ribonuclear proteins and the abbreviation for these is SNRNPs, sometimes casually referred to as SNRPs. And these contain catalytic RNA subunits. That are described as ribozymes. And they have catalytic activity within the spliceosome. And their function is for RNA processing. That's what the spliceosome does, is it processes RNA after it's first made. When RNA is first made in a eukaryotic cell, it's called primary RNA. But ultimately, primary RNA goes through a series of cuts and splices to become what's eventually called messenger RNA. So later in the genetics chapter, you'll hear about so-called messenger RNA, and it comes from the action of the spliceosome. And so we learn about this special class of RNA molecules that has enzymatic function. Many biochemical reactions involved in metabolism are referred to as redox reactions. And if we remember back to chapter 2 in chemistry with the acronym OIL-RIG, it stands for oxidized is lost and reduction is gained. And so an oxidation is when electrons then are removed from an atom or a molecule. And because matter is neither created nor destroyed, that electron has to go somewhere. And wherever that electron goes, there will be a gain of electrons, hence a reduction. And together, these coupled processes are referred to as redox reactions. And so they occur together as the electron moves from one place to another. Oxidation-reduction reactions are facilitated in part by coenzymes that act as electron carriers. In an earlier slide, in part 1 of this lecture, I talked about a list of coenzymes that function as electron carriers. And I'll start over here on the right with NAD, nicotinic adenine dinucleotide, which has a plus charge, and then a very similar molecule, NADP, which contains phosphorus and functions the same way as NAD, also has a plus charge, and then FAD, which stands for flavin adenine dinucleotide. And it doesn't happen to possess a formal positive charge, but all of these molecules can gain hydrogens and their associated electrons. And when they gain those electrons and hydrogens, they're said to be in a reduced state. And so NAD+, when it picks up a couple of hydrogens and a hydrogen and a couple of electrons, it will be written as NADH, and then also there'll be a proton left over that's lacking a hydrogen, And so most books, including ours, will write this reduced form as NADH plus H. And similarly, we can observe that NADP will function in the same way. So we can write the reduced form as NADPH plus an H plus left over. And the process is a little bit cleaner for FAD, where FAD is simply written FADH2. So both protons and both electrons that come with hydrogen as a pair will attach themselves to FAD. We can say that the reduced state in this column on the left is accurately described as energy rich because these extra electrons and hydrogens are present on the electron-carrying molecules. And in the right-hand column, we can say that without those extra hydrogens and electrons, that the molecules on this side are energy poor. Here's a very simple figure taken from our textbook that shows a redox reaction occurring. Here on the left, we have molecule A, which is a source of one or more electrons, which then can be donated over to molecule B that'll participate in a chemical reaction, in a redox reaction. Once A loses its electron, it will have entered into what would be referred to as its oxidized state. And we can see that as B then gains the electron, as an end result, B would accurately be described as being in a reduced state. Redox reactions are sometimes referred to, at least in part, as dehydrogenations when it comes to the oxidation of a molecule. So as hydrogens leave a molecule that becomes oxidized, then the electrons also travel with it. And so when you hear this term dehydrogenation, it's usually part of a redox reaction. This figure from our text more precisely shows a redox reaction as it's specifically facilitated by a molecule of NAD. So if you have an organic molecule over here that is a source of one or more electrons, and NAD plus can then participate in picking up those electrons and their associated hydrogens, then the way that this reaction will proceed is that the organic molecule will give up its electrons and hydrogens, and it will then be described as oxidized. And NAD plus, once it gains the associated hydrogens and electrons during this reaction, will have undergone a reduction and become energy rich. And it's written as NADH because, well, one hydrogen is shown here as a black dot, and then the two electrons that came from the H2 molecule will be situated as we see here, and then there will be a free-floating proton without any electrons floating next to it to account for all the matter in this redox reaction. The generation of ATP in a cell is most often produced by redox reactions. A series of redox reactions in the electron transport chain, as we'll see in some later slides, will bring about the synthesis of ATP. And synthesizing ATP comes from the cell using enzymes to obtain ADP, which stands for adenosine diphosphate, and then attaching a third phosphate group to that and to the ADP to make ATP, adenosine triphosphate. And the wavy lines that are drawn here are basically covalent bonds that have a high energy nature to them. So phosphate, which is one phosphorus and four oxygens, has many minus charges. When it's floating independently in a solution, it would be written PO4 minus 3. So we know that phosphates have a lot of negative charge. And when this negative charge comes in contact with into close proximity with other negatively charged phosphates, we know that like charges repel. And so these negative charges brought into close proximity with one another will cause a certain energetic repulsion. And so the bonds between the phosphates are denoted by a wavy line as containing a lot of energy, where if you were to take a pair of scissors and cut this, a lot of energy would be released because these negatively charged phosphate groups are repelling one another and they're held together by this one single covalent bond. So this is a high energy bond. And the more negative energy or negative charges that you add, like one here between these two phosphates, and then another one here, the energy actually goes upward in this direction. So as you add additional negatively charged phosphates, up to 3 here as a maximum on for ATP, then you get more energy. And so when we look at ATP structure, each one of these phosphate groups has an individual label or name. This is the alpha phosphate, and it has the least amount of energy. If adenosine has one phosphate, it's called adenosine monophosphate, or AMP. And then there is the beta phosphate here, and that has an intermediate amount of energy when it exists as adenosine diphosphate, ADP. And then we have the gamma phosphate here. And that third phosphate that represents 3 phosphate groups in ATP, the gamma phosphate and the bond that holds that on has the most amount of energy. By comparison, there are 3 different types of phosphorylation reactions that can bring about the generation or synthesis of ATP. And the first one we'll learn about is substrate-level phosphorylation. And a second type of phosphorylation event that some cell types can undergo is photophosphorylation. And specifically, these would be in photosynthetic organisms that can use the sun's energy to move phosphate in the cell onto ADP so that it can become ATP during photophosphorylation. And a third mechanism for phosphorylation to make ADP or ATP is oxidative phosphorylation. And this is where the electron transport chain and a series of redox reactions between electron carrying complexes will bring about the formation of ATP during what's called oxidative phosphorylation. Substrate-level phosphorylation can be described as a substrate that has a phosphate to donate. And it'll donate that phosphate towards the formation of ATP. Phosphates in this figure are abbreviated with a P, and a circle around it. And so that phosphate will be donated to ADP to form ATP. So in this figure, on this side of the equation, on the left side, the molecule, they're just showing a carbon skeleton of some sort, some kind of organic molecule. They're not showing the hydrogens there, but this chain of carbons, some molecule has a phosphate that's on offer, and so we refer to this part of the reaction as the substrate molecule. And this phosphate containing substrate will allow the phosphate to leave and travel over to ADP to make ATP. And that's really all substrate level phosphorylation is referring to. And sometimes students can be confused by this term, but it's no more complicated than that. A phosphate is available on a substrate. It can be taken from that substrate and put onto ADP in order to make the ATP as an end product. For this course, we'll keep photophosphorylation at a surface minimum level of description, and we'll just simply say that photophosphorylation converts light energy from the sun into ATP, which is a chemical energy in the cell, as well as an electron-rich NADPH molecule. A whole series of events are involved in photophosphorylation, and other biology classes that spend more time on photosynthesis will go into the details here. For our purposes, we'll leave it at this very brief description. Oxidative phosphorylation, which is found in all cell types, is associated with the electron transport chain and its chemical reactions involving a series of electron-carrying complexes, and then the formation of ATP at the end of this process. So we'll be looking at the specifics and in some figures on the electron transport chain, or ETC. There are two major modes of metabolism that can occur in most cell types. The first would be respiration, and respiration comes in two major types. There can be aerobic respiration, which involves the use of oxygen, molecular O2, or if oxygen is not present or available, some organisms can make the use of anaerobic respiration by using molecules such as sulfur, or I should say sulfate, or carbonate, or nitrate. And although they contain oxygen, they are not O2, and so they're classified as forms of anaerobic respiration. Alternatively, another type of metabolism is fermentation, and fermentation is also anaerobic, but it's not respiration. Technically speaking, it is a different type of metabolism altogether. One of the most important defining characteristics of respiration as a form of metabolism is the inorganic end products that respiration produces. CO2 is inorganic, and so is H2O, because neither end product molecules contain both carbon and hydrogen. The final electron acceptor, or FEA, involved in respiration is either oxygen, as in O2, as a final electron acceptor, if it's aerobic, and then anaerobic forms of respiration may use other molecules besides O2, such as sulfate, or carbonate, or nitrate. And although sulfate, nitrate, and carbonate contain oxygen, they are not actually O2, as in molecular oxygen. So, as we look here at aerobic respiration, we can also take into account that this is the type of respiration or metabolism that forms the most amount of ATP per molecule of glucose. Now, if we reference some older numbers taken from our book that have not been updated, even in the latest edition of our textbook says that either 38 or 36 molecules of ATP may be yielded as 1 molecule of glucose is aerobically processed. So we'll say that this is in the book, that these are old numbers. More recent research papers and sources show that this number has been revised downward to 30 to 32. And so I'll write that down. 1 glucose, when aerobically processed through respiration, yields either 32 or 30. And so we still see 2 molecules of ATP in the difference between 32 and 30, and specifically this refers to the difference between prokaryotes and eukaryotes. In prokaryotes, 32 ATP may result as a maximum yield from 1 glucose molecule processed. So the bacteria, the prokaryotes, can make a couple of more ATP molecules than their eukaryotic counterparts. And what accounts for this is transport across the membranes of the mitochondria. So eukaryotes have membrane-bound organelles, including mitochondria, and the mitochondrial membrane and transport across that membrane requires a couple of extra ATP molecules. So for the purposes of our exam, I will use the more accurately revised numbers that are more recent than the books. But any way you want to look at it, we can say that whether you're using the older numbers from the book or the newer numbers that I've presented here, these numbers are still the most amount of ATP that an organism would yield after processing 1 glucose entirely through the aerobic respiratory process. When comparing anaerobic respiration to aerobic respiration, there are some commonalities because it's still a form of respiration, such as the inorganic end products. So CO2 and H2O are both inorganic, and those end products may result from anaerobic respiration. Anaerobic, though, by definition means that this form of respiration happens without O2 specifically. So molecular oxygen is not used, and instead anaerobic respirers have been found to use various molecules such as sulfate or carbonate or nitrate. And although these molecules contain oxygen, they are not considered molecular oxygen as in O2. And so it still qualifies as anaerobic. And anaerobic respirers have been found to be slightly less efficient than their aerobic counterparts. So we'll make a note here that slightly less ATP is yielded. So when a molecule of glucose goes in, the range is somewhere between 3 to 29 ATP per molecule of glucose. 3 seems pretty low on the low end, but there are so many different species and examples of anaerobic respirers that have been studied that there can be quite a range. However, this range is always going to be at least somewhat less, even if it's only 1 molecule of ATP less than the aerobic respirers by comparison. Now we can compare fermentation to respiration. By contrast, let's make sure that we're clear. This is not anaerobic respiration. Many sources on the internet and various textbooks have sometimes described fermentation as a form of anaerobic respiration, and this is an incorrect application of the concept anaerobic respiration. Fermentation is specifically different in one major way, which is that it results in organic end products that contain both carbon and hydrogen. So there's one major difference. And a commonality would be the anaerobic nature. There's no molecular oxygen used during the fermentation process. Also, fermentation is very much inefficient by comparison to respiration as a process because glucose is incompletely broken down. It is not fully oxidized, and the available energy that glucose has to offer is not completely extracted. So fermentation as a process will only yield 2 ATP, and this then characterizes organisms that are slow growing if they're undergoing fermentation as their means of growth. And there are 2 major types of fermentation that we'll describe here in the coming slides details wise. We have lactic acid fermentation, and this is a type of fermentation that happens in the human body, but also there are some bacteria that are lactic acid fermenters. And we also have alcoholic fermentation. And so some species of organisms will perform alcoholic fermentation as opposed to lactic acid fermentation, which is a different process. And these modes of fermentation are based on, their names are based on what is produced as end products. Now that we've looked at the general summaries of what defines aerobic respiration and anaerobic respiration and how those forms of respiration compare to fermentation, we should dive into the specifics of aerobic respiration as a process. And before we do that, let's take into account the overall equation involved in aerobic respiration. This equation starts with a molecule of glucose, and the molecular formula indicates glucose has C6H12O6, and when 1 molecule of glucose goes in, it is combined with 6 molecules of oxygen, O2, and after the respiratory process is complete, 6 molecules of carbon dioxide will be yielded along with 6 molecules of water, and the big E is for energy. And so now we'll go through the 4 major steps, A through D, that are involved in aerobic respiration. Glycolysis is the first step in aerobic respiration. And here is a very simplified figure taken from our text that shows 1 molecule of glucose entering in, and then we'll label that 2 pyruvic acid molecules, also known as pyruvate, come out, and we have 2 molecules of ATP that will be netted total out of this process, as well as 2 molecules of NADH. This simplified figure is leaving out that there are actually 10 steps that are contained within this arrow, starting from the beginning to the finish here. So in summary, 1 glucose enters into glycolysis. Glyco refers to the sugar glucose, and lysis means to break it apart. And when that glucose is completely broken down, or completed through the glycolytic process, we'll see these products yielded. Taking a look at glycolysis in a little bit more detail, we can see the 10 steps split into the next 2 slides. This slide shows the first 5 steps, and for this course, we will not be required to memorize each step and intermediate that's involved, but we can at least say that the first 5 steps of glycolysis are referred to as the preparatory stage, also known as the energy investment phase. So the first 5 steps are not something that generate excess energy for the organism. This is a time when the organism is performing these steps so that there's a payout during the second 5 steps. And so we can see in these first 5 steps, called the energy investment phase or preparatory stage, that 2 molecules of ATP go in, so they're used by the process here at this very first step, and then at the third step another molecule of ATP is used, and so ATP is donating its phosphate to become ADP, and the phosphate group is attached to glucose here, and then again down here when glucose is rearranged into fructose at some point along the way here. Ultimately though, this will then be split into 2 3-carbon containing molecules, and then we'll move on to the final 5 steps. In the final 5 steps, we'll refer to this as the energy-conserving stage. Another way of looking at this, another way we could describe this would be to call it the energy-gaining phase. So this is where the payout happens energetically. And overall, by the time all 10 steps are complete, glycolysis will yield a total of 2 ATP. So in the previous slide, with the first 5 steps, we saw 2 ATP used, but then during these last 5 steps, we can see that 2 ATP are yielded here, and then 2 more ATP are yielded here. So if a total of 4 ATP are made during these final steps of glycolysis, and we minus out the 2 ATP that were invested during the first 5 steps, we get a total yield of 2 ATP. For the purpose of this course, in this section, we'll just make sure and keep these facts in mind. 1 glucose goes in to glycolysis, 2 pyruvate or pyruvic acids come out. Also, 2 molecules of ADP go in, and then 2 molecules of ATP come out. And we can say that 2 NAD pluses go in, and after they gain hydrogens and electrons, 2 NADHs total will come out. Following glycolysis, the intermediate step occurs, and sometimes, in some books, will describe the intermediate step as the transitional step. So another name for it can be the transitional step. And what this step involves is that the products from glycolysis enter in. So a pyruvic acid enters in, and we'll make this as a column like the last slide. What goes in is pyruvic acid or pyruvate, and what comes out is something called acetyl-CoA. Additionally, we can say that the intermediate step requires one NAD plus to go in and one NADH will come out. When we look at this process, we should also be aware of the number of carbons that go in. Pyruvic acid is a 3-carbon molecule represented by these blue circles here, and this 3-carbon pyruvic acid will undergo a decarboxylation, and CO2 will be released as a gas, and the 3-carbon pyruvic acid will ultimately become a 2-carbon acetyl group. Meanwhile, a coenzyme A is attached to that, so ultimately the end product is acetyl-CoA. So we should write that down as well, that a molecule of CO2 comes out when the 3-carbon pyruvate is trimmed down into a 2-carbon acetyl-CoA. The reason for the intermediate step is in preparation for the Krebs cycle, to feed the Krebs cycle. And so here we can see the acetyl-CoA that was produced during the intermediate step as it enters in at the top of the Krebs cycle at step 1. The Krebs cycle goes by many different names. So don't be confused if you also hear of the Krebs cycle referred to as the TCA cycle. TCA is a technical name for, is an abbreviation for tricarboxylic acid. And that's just another name for citric acid. So tricarboxylic acid is also known as citric acid. So another name for the Krebs cycle is the citric acid cycle. So pictured here, where acetyl-CoA goes in, we see citric acid form, and it is a 6-carbon molecule represented by the 6 blue circles here, right after step 1 that says citric acid. So citric acid forms after the first step of the Krebs cycle, and then there are many steps involved and many intermediates that are formed during this cyclical pathway. For the purposes of this course, we will not be required to memorize all of the intermediates and the steps and the enzymes that are involved. Rather, what I would like you to be aware of are some of the major end products that are gained out of the Krebs cycle, so that we can learn its basic purpose. So let's keep track here a little bit. We can see an NAD molecule goes in, and an NADH comes out. So NAD picks up some hydrogen and some electrons and gains some energy. We can see this process happen at another step as well. And even at another step one more time towards the end of the Krebs cycle. So the Krebs cycle generates 3 molecules of NADH, and also, if we look closely, we see that there is an FAD, electron-carrying molecule, that picks up hydrogens and electrons as well. So during this step of the Krebs cycle, we see a molecule of FADH2 yielded. Additionally, we see that a molecule of ATP can result from the action of the Krebs cycle. And so the Krebs cycle has many steps where after citric acid forms, transformations of the carbon skeleton occur and some carbon is lost. We can see here a molecule of CO2 comes out, and yet again another molecule of CO2 will come out at a different step. And so every time the Krebs cycle turns, some CO2 is made, and this accounts for part of the CO2 that we exhale or we produce during the respiratory process. So looking at the Krebs cycle qualitatively, we can say that we should definitely keep in mind the main purpose of the Krebs cycle after looking at these major products. So the main purpose of the Krebs cycle is to generate the majority of energy-rich electron carriers. And what are the electron carriers? So if we recount, that will be NADH and FADH. And in the next slide, we'll keep count on what goes in and what comes out as a summary. Here we have the summary then. What goes into the Krebs cycle initially is acetyl-CoA, and this 2-carbon molecule with the coenzyme A attached, as it enters into the Krebs cycle, we know that it forms citric acid. But ultimately, the 6-carbon citric acid that goes in will be decarboxylated twice as it changes forms, and we know that a couple of molecules of CO2 come out. We also know that 3 NAD pluses must enter into the Krebs cycle in order for 3 NADHs to have picked up their hydrogens and electrons to be produced. Also, 1 FAD goes in, and what comes out is an energy-rich FADH2, and 1 ADP goes in and picks up a phosphate and becomes ATP. So every time the Krebs cycle turns, these items go in and these items come out. Keep in mind, however, that the Krebs cycle will turn twice for every glucose that enters in. If you recall back to the glycolysis step of respiration, glycolysis splits the glucose into 2 molecules that eventually become pyruvate. And so if there are 2 pyruvates or pyruvic acids, then those go through 2 intermediate steps, and then acetyl-CoAs come out, then there'll be 2 acetyl-CoAs, and each acetyl-CoA will turn the Krebs cycle once. So the Krebs cycle will turn twice, and we would say then that the Krebs cycle generates 2 ATP from 1 molecule of glucose. And the way that that ATP is generated is through a substrate-level phosphorylation event. The 4th and final step of aerobic respiration is the electron transport chain, or ETC. The electron transport chain is located in either the inner membrane of the mitochondrion, if we're looking at a eukaryotic organism like the human, or in bacteria, prokaryotic organisms, house the electron transport chain in the plasma membrane or inner cell membrane of their structures because they lack membrane-bound organelles. They do not have mitochondria. ATP is formed through a process referred to as oxidative phosphorylation. And this oxidative phosphorylation will be carried out by an enzymatic complex called ATP synthase. And preceding this event, electrons will move through a series of electron-carrying complexes. So I'm going to cross out the word carrier here, only because I don't want anyone to think that carriers is referring to NADH and FADH2 as we previously talked about electron-carrying molecules. Here we're referring to electron-carrying complexes. And they're labeled complex 1, complex 2, 3, and 4. And these electron-carrying complexes are housed in the electron transport chain. And so we'll see that figure in the next slide. Here's an illustration of what the electron transport chain looks like. Complexes 1 through 4 are not officially labeled here, but are actually illustrated with their technical names. So FMN means flavin mononucleotide. The Q here stands for quinones or quinones, and then CYT stands for various cytochromes. And so instead of memorizing what these complex chemical names are, we'll just qualitatively take a look at this. Complex 1 here, complex 2 here. This is complex 3. This little bubble here that's labeled cytochrome C is not officially one of the complexes. Complex 4 we would be most familiar with in reference to lab, where we learned about the oxidase enzyme, which is also referred to as cytochrome A3. And so looking at this overall, we can say that NADH, an electron-carrying molecule, will drop off hydrogens and electrons, and the hydrogens separate from the electrons. And they're pumped through the membrane and into periplasmic space, or above the inner mitochondrial membrane. And so right here, hydrogens build up, and if we could take a look at that, this would be defined pH-wise as an acidic space. So we'll look at that in just a moment, but acids are building up here because of the protons. Meanwhile, electrons are received and are passed from the first complex over to the second complex, third, and fourth, until eventually those electrons are dumped back into the area below the membrane, where they can recombine with hydrogens, which ultimately are flowing through this ATP synthesizing complex next door to the electron carrying complexes. So ATP synthase physically, mechanically rotates around as hydrogens flow through an area from high concentration to an area of lower concentration. This phenomenon is driven by what's called an electrochemical gradient. And this is in reference to describing all of those hydrogens that build up. So an electrochemical, and it's a gradient because there's an area of high concentration up here above the membrane, and it flows towards an area of low concentration below the membrane. And there are other names for this as well. Another way of describing this, aka also known as the proton motive force. And this is because protons are building up, and there will be a physical motive or pressure that acts as a force that can then be used to drive the ATP synthase complex to rotate and to pick up a molecule of ADP and join it with a phosphate in order to make ATP. And another way of describing this is to say that this entire event is driven by chemiosmosis. So these are all technical terms that are used or applied to describe the same phenomenon. So chemi is referring to chemicals. Osmosis is referring to a pressurized event here where we have more hydrogens on one side of the membrane flowing towards an area with fewer. And this event is set up by the proton pumps that are pumping protons across the membrane. So complex 1 can act as a proton pump, and coincidentally so can complex 3. And so we can see protons pumping through these areas. We don't have to memorize for this course which complexes are proton pumps, but at the end of this process, we can keep in mind that the major characteristic is that all of these protons or hydrogen ions are building up, and this area is very acidic and is in a high concentration that will drive this process ultimately to form ATP. Let's summarize what goes in and what comes out of the electron transport chain. Overall, we can say that this is a continuous process. So rather than assigning absolute numbers here, we're just going to say that NADHs go in, and so do FADH2s. These are the energy-rich electron-carrying molecules, and when they drop off their protons and electrons, then NAD+ comes out, and FAD comes out. And also we know that ADP goes in, and once this process is complete, ATP will come out. That's the major goal of the electron transport chain, is to make large amounts of ATP. We know that oxygen is necessary for this because oxygen acts as the final electron acceptor. And when oxygen accepts electrons and the hydrogens that are associated with those electrons, ultimately water comes out. And so these are the molecules that go in and come out of the electron transport chain that we need to keep in mind for the purposes of this course. Anaerobic respiration, by comparison to aerobic respiration, is mostly similar. It involves glycolysis, and after the glycolytic step, it'll have the same intermediate or transitional step, but the differences begin with a shortened or stunted Krebs cycle, and also an electron transport chain that may be abbreviated to some extent as well. And so that's why anaerobic respiration results in fewer ATP formed as an overall process when glucose goes in. And it varies by species, so the number of ATP that's reduced depends on how shortened the Krebs cycle is or how abbreviated the third and fourth processes are in the Krebs cycle and electron transport chain. Let's take a moment to keep track of where respiratory processes occur in both eukaryotes and prokaryotes by comparison. Glycolysis as a pathway occurs in the cytoplasm. And this is true regardless of eukaryotic or prokaryotic life. The cytoplasm is the place where glycolysis occurs. The intermediate step, however, can occur in different places. The intermediate step is described as occurring in the mitochondrial matrix. This is the innermost part of the mitochondrion in eukaryotic cells. And since prokaryotes, the bacteria, do not have mitochondria, we say that the cytoplasm is the place where the intermediate step occurs. The Krebs cycle comes next, and this also occurs in the mitochondrial matrix of eukaryotic cells. And once again, prokaryotes lack mitochondria, and so they perform the Krebs cycle in the cytoplasm. For the last step of respiration, the electron transport chain is occurring where it's housed in the inner mitochondrial membrane for eukaryotes. And because there is no mitochondrion in the prokaryote, it is the inner cell membrane, or cytoplasmic membrane. This figure taken from our text is a comparison that shows how respiration and fermentation differ side by side. And we see right away that respiration has more steps, beginning with glycolysis and then going through the intermediate step, which isn't labeled here, but then the Krebs cycle, and finally the electron transport chain occurring during respiration. By comparison, fermentation also makes use of glycolysis, and the product of glycolysis, the pyruvic acid, is fed into a unique process where organic end products form, as opposed to the inorganic products of water and carbon dioxide. We see that fermentation will form things like lactic acid or alcohol. We've gone over the specifics of respiration, and now we can examine fermentation in a little bit more detail. Generally, fermentation can be referred to as any spoilage of food by microorganisms. Sometimes people will say that in industry or in casual terms when talking about fermentation. The dairy industry will talk about fermentation when making yogurt or certain cheeses, and the brewing industry, whether people are making wine or beer or other alcoholic products, they'll talk about fermentation in a general sense. And some parts of industry will just simply call it any large-scale microbial process where air is absent. And so these are very common but somewhat inexact definitions for fermentation. So here are the precise characteristics of fermentation, and we can compare them to anaerobic respiration and see how they're similar and how they're different. First, anaerobic fermentation is without the use of oxygen, just like anaerobic respiration. However, fermentation only yields its ATP from substrate-level phosphorylation. Specifically, the ATP come from glycolysis, as all fermenters get their energy yield from the glycolytic step. And the final electron acceptor is unique for fermenters in that it's an organic molecule that's involved. It has to be a final electron acceptor that contains both carbon and hydrogen. When we compare that to respirers, whether they're aerobic or anaerobic respirers, the final electron acceptors are inorganic molecules like oxygen, nitrogen, nitrate, carbonate, sulfate, etc. Fermenters are said to start their process with the end products of glycolysis, namely the pyruvate, as we'll see. And fermentation is also characterized by being an incomplete oxidative process of glucose. So only some of the energy that glucose has to offer will be yielded by the fermentation process. And keep in mind from an earlier slide, there are two major types of fermentation. And they're named after the end products that they make. The first type is lactic acid fermentation. And the second type is alcoholic fermentation. When we compare lactic acid fermentation on the left to alcoholic fermentation on the right, there are some commonalities and then some differences. The common part is the glycolytic step, where glucose is split ultimately into two pyruvic acids or pyruvates. But then, where the pyruvate ends up, or how it's handled, is how these two fermentative processes differ. So with lactic acid fermentation, which is a bit simpler fermentative process, there's really only one major final step left, which is that the pyruvate acts as the final electron acceptor. So the FEA is pyruvate. And when lactic acid forms, it's due to the fact that the pyruvate receives electrons from NADH. So NADH will give up its hydrogens and associated electrons, and it will become NAD plus again, so it can recycle into another process somewhere else in respiration. And these hydrogens and electrons, as they attach themselves to pyruvate, will end up arranging into a molecule of lactic acid. And by comparison, when we look at alcoholic fermentation, we can see that the pyruvic acid initially undergoes a decarboxylation. So this 3-carbon compound will become a 2-carbon intermediate compound called acetaldehyde. And acetaldehyde is ultimately the second to last step, where it acts as the final electron acceptor during alcoholic fermentation. And at the end of this process, ethanol will form, which is known as drinking alcohol. So alcoholic fermentation differs in that it has one more step, and it forms CO2, and its final electron acceptor is different. Final electron acceptor is acetaldehyde. Lactic acid fermentation can come in a couple of different forms. We know that the pyruvate is reduced by NADH and lactic acid forms, but different species of lactic acid fermenters can be described as either homolactic or heterolactic. Homo meaning same, and what it means ultimately is that only lactic acid is produced as a product. So lactic acid only. And many homolactic fermenters are used in industry to make common fermented milk products, like yogurt would be the most common. And some types of cheeses, like cottage cheese. And speaking of cheese, some more complex cheeses that have interesting, kind of unusual flavors may involve the use of heterolactic fermenters. And these types of fermenters will produce lactic acid, but then also other acids. So lactic acid and other acids. A common example of this would be an organism that makes Swiss cheese. So Swiss cheese has an unusual, unique flavor by comparison to most cheeses. And what's responsible for that unusual, sharp flavor that Swiss cheese has is that another acid called propionic acid is formed. And it's from a heterolactic fermenter that it's involved in its production process. And also, Swiss cheese has giant holes in it, and those holes come from gas. We know that some lactic acid fermenters also can produce large amounts of gas. As a final summary slide to end this chapter, let's fill out a table where we can keep track of respiration and fermentation as it's broken down by events. We know that glycolysis overall will yield 2 ATP net, and we know that glycolysis precedes all modes of metabolism, including aerobic respiration, anaerobic respiration, as well as fermentation. And therefore, we can say that glycolysis does not require oxygen. And when glycolysis generates ATP, it is through a substrate-level process. And there really is no finality to glycolysis. It is just a step that leads to other steps in metabolism. So we're not going to talk about a final electron acceptor in glycolysis. The Krebs cycle also can produce 2 ATP, but that's for taking into account 2 turns of the Krebs cycle for a molecule of glucose going in. And oxygen is not directly required by the Krebs cycle. And when those ATP form, as the Krebs cycle turns, this is also produced by a substrate level event. And the Krebs cycle has no finality, no final electron acceptor. So we'll just put a dash mark here. And the electron transport chain produces by far the most amount of ATP. And we'll go ahead and put 28 ATP here, but it depends on the organism. So going with the modern numbers, it'll either be 26 for prokaryotes, or eukaryotes, sorry, or 28 for prokaryotes. And oxygen is required. Remember, O2 is the final electron acceptor in aerobic respiration. And the phosphorylation events are oxidative. So a series of electron-carrying complexes will pass electrons through it, and oxidatively ATP will be formed. And we know the final electron acceptor is O2 for aerobic respiration. Now, for the column here that says, or the row here that says anaerobic respiration, we're going to take this as overall. So all events involved in anaerobic respiration, including glycolysis and whatever the Krebs cycle would be. And we'll say that the number of ATP ranges. And earlier we had mentioned that it could be anywhere from 3 to 29 ATP total. Okay, total. And oxygen is not required by definition. It is called anaerobic. And since we're talking about anaerobic respiration overall, including all steps, we'll say that this is a combination of substrate for the glycolytic and abbreviated Krebs cycle steps, as well as the electron transport chain, which is oxidative. And the final electron acceptors varies by species. So some use nitrate, and other species can use sulfate. And other species may use carbonate. And these are common examples. There can be other molecules not mentioned here that might be used by less usual or unknown anaerobic respirators that are out there in the environment. And lastly, we can compare fermentation to these respiration processes and say that the ATP is a yield of 2, and that actually comes from the glycolytic step that precedes the fermentation processes. And we know that by definition that fermentation is not a respiratory, it's not an oxygenated event, and so no oxygen is required. And the ATP that's produced is substrate level, just like it is in glycolysis. Substrate level event occurs, and then we can look at which style of fermenter we're talking about. If we're talking about a lactic acid fermenter, then we can say the final electron acceptor is the pyruvate, which is directly used to produce lactic acid as a final end product. So pyruvate can be the final electron acceptor, or if we're talking about an alcoholic fermenter, then the final electron acceptor is acetaldehyde. And this concludes our coverage of Chapter 5 on microbial metabolism.